So the aim of the background is to impress upon you why somatic variant discovery needs different tools and different workflows than the germline context. With somatic variant discovery, we want to detect changes against germline variation. So each of us are born with our germline variation, and as we age, we accumulate somatic mutations from sun exposure, from smoking, from errors in DNA replication and repair. And some of these somatic mutations can turn malignant and cause cancer. And typically in our workflows, when we talk about somatic variant discovery, we're talking about looking at cancer genomes against germline variation. However, our workflows can be applied to other somatic contexts, or for research instances where you want to call for variable ploidy, for example, mitochondrial variant calling. So many cancer alterations could be said to be passenger mutations that aren't really driving the cancer progression. And only some of the mutations are driver mutations that you want to find out what they are, because they are what are going to inform treatment, for example, in personalized medicine. And so the sample paradigm in cancer is a matched set, a tumor with a matched normal from the same individual, because we want to account for germline variation from the same individual. And in many ways, every cancer genome is unique, like a snowflake. And different cancer types can converge to look like each other. And these are reasons why we use large cohorts, large sample numbers to get to the driver mutations. I was trained as a biochemist myself, and this is how I think of cancer genomics. Determinants of cancer include genetics that contribute to risk encoded in our germline DNA. That's at the top of this tree, and also include non-genetic factors that contribute to mutations, like environmental factors, which I show at the bottom of the tree. So the high penetrance genes that we think about, like BRCA1 and BRCA2 in breast cancer, these account for only 5% or less of cancer. The drivers are below. So certain cancers are more prevalent in certain sexes. For example, for 2015 in the United States, there were 3.2 times more men diagnosed with bladder cancer than women, and 2.6 times more men who passed away from bladder cancer than women. And finally, you know that viral infections can lead to certain cancers. Does anyone know what this particle might represent? Any virologists in the house? No? Oh, well, this is HPV. Yep, that is correct. And it causes, yes. And so there is now a vaccine against HPV, and I actually was part of the clinical trials. I know I've been immunized, and I have reduced my risk of cervical cancer. Here are two reasons why cancer genomics calls for increased sensitivity. One is contamination, and the other is heterogeneity. When I say contamination, I mean normal contamination in a tumor biopsy sample, or tumor contamination in the normal biopsy sample. And so this is actually something we refer to as tumor purity, where it's defined as the number of tumor cells over the total number of normal and tumor cells. So tumor heterogeneity is something I will explain with the chart below. You've seen this before. Here we have an illustration at the bottom, progression in time from left to right, where we're accumulating more mutations with darker colors. And with the purple and blue, we find from pre-malignant to malignant tumors. In these circles, we're showing two different pathologies of the tumor. One is called segregated and the other intermixed. Either way, when you sample the tumor, you're going to get additional subpopulations that have different mutations on top of the clonal cancer population. And so we have to rely on our pathologists and algorithms to sort these kinds of factors out. Here's another view of cancer evolution. Here we have a normal epithelium that starts to accumulate mutations. Each of these colors are a mutation, and that divide the population of cells into distinct populations. A lot of these mutations are passenger mutations, and some of them are driver mutations that are driving the tumor forward to malignancy. And so when we take a tumor biopsy, this is what we're getting, a very heterogeneous population. And so we need increased sensitivity to be able to detect things that might be at very low allele fractions. One last thing. So here at the end, what has gone awry are pathways for apoptosis, DNA repair, and also immune clearance of cancer cells. That's another component of cancer that researchers are interested in these days. So purity and heterogeneity contribute to the noise. And so the amount of signal can be comparable to the noise for somatic analyses. And this is different from the expectation for germline studies, where we have an allele fraction that we expect to follow ploidy assumptions. If you have a diploid organism that has a heterozygous site, half of the pileup we would expect to be one allele and the other half to be another allele. And this signal is much greater than the noise. For somatic variants, we have on top of our noise purity and heterogeneity issues, and we cannot rely on ploidy assumptions because a lot of things can go wrong in a tumor cell, including aneuploidy. So it becomes a real challenge to suss out what is going on. Here are some cancer-specific challenges that I've listed. We have all of the challenges of germline sequencing plus additional challenges. And I'd like to point out that two of them we have discussed, purity and heterogeneity, plus this third one, cross-sample contamination, increases our need for sensitivity. And cross-sample contamination, I mean that the tumor sample will have low levels of another individual's DNA in the sample. And this might appear like a low allele fraction tumor subpopulation, but in fact it's contamination. And mistaking that for somatic variants would be not good for your analysis. Here, lastly, we know that cancer has a wildly different mutation spectrum than the germline variants that we observe. For example, we have chromothripsis, which is like a bomb has gone off in the nucleus. All your DNA is fragmented into little pieces and then randomly religated together to in a process called chromothripsis. We have aneuploidy, which are chromosome number abnormalities, copy number alterations, structural variants, which are large variations, SNVs and indels, and something called loss of heterozygosity, where you can lose the variation but maintain the copy number level. And we'll get into that at the very end a little bit. And so all of these different types of mutations call for different types of tools in the somatic field. And so if we increase our sensitivity, what else do we increase? You answered this for Yossi? Yes, we increased our false positives. And so with the increased sensitivity, we need specialized filters to make sure we remove these false positives. And these false positives come from two major sources, artifacts and germline variation. So let's focus on the left-hand side. Here we have a tumor and a matched normal, and we have these blue mismatches that appear like a somatic mutation because we don't see it in the normal. However, it turns out that this is an artifact of sequencing because if you look closely, each of the reads that support this variant are in a single direction. And so we want to remove these types of artifacts, and we have filters in place for that. This type of artifact can strike at any base, and the source of these are misread bases, sequencing artifacts, and misaligned reads, mapping errors. And besides the filters, we can use something called a panel of normals to filter these out, and we will get into that in a bit. On the right, we have a seemingly somatic event because there are a lot of mismatches in red here and not so many in the normal. There's one read that supports the mismatch in the normal, and you wonder if this could be tumor contamination of normal, or if it is actually a germline event that we are unable to call because we have low coverage in this area. So having low coverage for the normal is a typical design in somatic studies because you want to spend all of your money sequencing the tumor at very high depth. So you sequence the normal at a lower depth. And so this kind of case occurs, and at risk are 1,667 germline variants per mega-base pair. And if you compare that to the average SNP or point mutation we expect for the average cancer, 1 per mega-base pair, I mean, there's a lot of signal that could be false positive. And we deal with these kinds of events by telling our tools what sites are commonly variant using, say, dbSNP, exact, the panel of normals, and these days, Nomad. Nomad is a very large data set of common variation, I think 20,000 exomes and 20,000 genomes and 120,000 exomes. That's a lot. That's a lot of data. So you can get allele-specific frequencies from that sort of data. So I want to wrap up the background section with a example from real cancer data. Here, this, and it looks complicated, but I'm going to walk you through it. I have done something. Where did this, let's see. Preview, there we go. So this is a plot that's made from a brain cancer cohort. It's called an iCommute plot from firebrowse.org. And you can actually go in and browse their data and make such plots for whatever cohort you are interested in for this project. And you can see that they've subdivided the cohort into some different stages of the brain cancer. So each column represents a cancer patient. And here we have mutation significance. These significantly mutated genes are sorted from most significant to least. And you can see that many of these patients have the same mutations in the same loci, namely IDH1 and 2, TP53, and this gene called ATRX. But there are a number of patients for each of these subgroups that don't have mutations in these genes. And so if you look at their copy number gains and losses, you see that they have increased focal copy number alterations that are enriched for each of them for the same regions. And this just goes to show that cancer analyses has to take into account a wide spectrum of alterations, and you can't just do SNV and indel calling. You must also consider copy number variation. So we're going to go through two workflows: short variants with somatic SNV and indel, MUTEC2, and that's on the left, and somatic copy number alteration detection on the right. So I just want to note that the MUTEC2 workflow here on the left is still in beta status. We're still changing things and working out some optimizations. And the CNV workflow is ready for your use for targeted capture data. For whole genome seq data, we're still developing the workflow and the parameters. Oh, and one thing to mention, and I think Yossi brought this up, is that the pre-processing for the cancer samples is pretty much the same as the germline, except we do not do binning for BQSR. So if you want to see what status a tool is in GATK4, you can use GATK-launch, which invokes the jar. Use --list. You'll get a list of tools and then a description of the tools alongside their status. For example, here it says beta tool experimental, which means basically it's an alpha status. You can use it if you'd like, but things will change in the future. Results may not be guaranteed. You should be careful what you are using. Of course, we'd like for you to try everything and give us some feedback because we do depend on feedback from the user community. So for those of you who are familiar with GATK3, some differences in GATK4 are that we repackage functionalities. Basically, we take functions in GATK3 and we put a different tool name on them, or we break them out into two different tools and call them something different. But the underlying functionalities are pretty much the same. It's just the surface packaging is different. For example, to estimate cross-sample contamination, in GATK3 we have contest, and in GATK4 we have two new tools, get pile-up summaries and calculate contamination. To create a panel of normals for short variants, in GATK3 we use MUTEC2 and combined variants. In GATK4 we use MUTEC2 and a new tool called create somatic panel of normals. And in both versions of GATK, to call somatic short variants we're using MUTEC2, but they are different. One of them capitalizes their T and the other has a lowercase t. And that's to make sure you're using the right tool. Like your scripts will fail because the tools are actually pretty different. One thing that is different is that before filtering was built into the MUTEC2 caller, but now we have separated out filtering into a separate tool called filter MUTEC call. And we have a new filtering functionality for sequencing context. And this tool is called filter by orientation bias. And eventually we have plans to merge this into filter MUTEC calls, but right now we're testing it, so it's a separate tool. So just to reiterate, beta tools are in active development. Do not use them on your clinical samples. Do not use them in production. Feel free to play around with them and let us know what you think. And I have to say, even though both versions of MUTEC2 has been in beta status, the Broad Cancer Genome Analysis Group has used GATK3 MUTEC2 to call indels and has published that data in peer-reviewed journals. Okay, that's it for the background. So let's go on to SNV and indel discovery. This is again with MUTEC2 and filter MUTEC call. And all of these tools I'm talking about will be in GATK4. So as I said, the annotation functionality is separate from the filtering functionality. MUTEC2 is what calls and provides the annotations in this left-hand diagram. It takes a tumor normal match set and optionally takes a panel of normals, as well as optionally a germline population allele frequencies file. meaning it has allele-specific frequencies for germline population variants. And all of these go into MUTEC2 to give us a raw SNV and VCF. There is no math here. This is a VCF format file. And this goes into our filtering tools alongside metrics, some of which were optional. For example, this collect pre-adapter metrics. You may not need it, but if you do, you just need to collect some metrics with our tools, as well as the metrics from estimating contamination. And you filter your variants to get analysis-ready somatic calls. So filtering is just represented very simply, but there's a lot going on in filter MUTEC calls. And these specialized downstream hard filters allow us to have higher upstream sensitivity. Having tried both GATK3 MUTEC2 and GATK4 MUTEC2, I have to say it's much faster. And if you ask our developer about it, and I should say the two main people who work on this are David Benjamin and Takuto Sato in the group. And their office is right next door to mine, so it's easy to go ask questions. It skips sites that are in the PON, and it skips sites for high fraction alt alleles in the normal, and only assembles sites where the possibility of being somatic is reasonable. And before we called mostly on the site level, it wasn't so pertinent that we have the actual alleles. But now MUTEC2 is allele specific, and it distinguishes between alleles in the germline population frequency resource and uses that allele frequency in calculating the probability that the variant exists in the normal and the tumor. One thing to note is that MUTEC2 does not call loss of heterozygosity events. So for example, if you have a multi-SNP site, MUTEC2 will not call an alt that was in the normal that disappears in the tumor. If the tumor is ref, but is still different from the normal, that's not called. We have a separate workflow to call loss of heterozygosity events, and we will touch upon that in the last 10 minutes. So underneath the hood of MUTEC2 is the HaplotypeCaller assembler. You should be familiar with the 4 stages from Laura's talk. Just to summarize, we identify active regions. Remember, we skip sites from the PON and sites in the normal that have high allele fraction alleles. We then assemble reads into possible haplotypes. And there is some pre-filtering that happens, and the pre-filtering for the tumor and normal reads are identical. MapQ greater than or equal to 20. Each read is aligned to each haplotype and assigned likelihoods via PairHMM. And we do genotyping in a funny way because we don't have an explicit ploidy assumption. We genotype via allele fraction. So again, somatic genotypes are inferred from PairHMM likelihoods. There's a Bayesian equation that I'm not going to get into because Laura has covered this fairly well. One thing to point out is that there isn't a ploidy assumption, and that there is a variable here that is a random variable that our developer says marginalizes over, that the tool marginalizes over using variational Bayes. It's just, in essence, it's just the math is more rigorous. And the statistical threshold for somatic calling uses log likelihood ratios greater than or equal to 5.3 in favor of the variant somatic genotype. And we also filter based on the likelihood of the alleles in the normal. So just like with HaplotypeCaller, we want to convert likelihoods into probabilities. And towards this, we use the population germline allele frequencies And this F that is representing the allele frequencies is different from the F alt from the previous slide. In this example, we are referring to the human nomad resource. And the tool follows this logic. If the variant is in the nomad resource, you use the allele frequency provided by the resource. If the variant is not in the nomad resource, it uses a default parameter, which we recommend that you change to represent the rarity of something that you wouldn't find in your resource. And a good rule of thumb is to set the allele frequency for a variant that's not in the resource to 1 over 2, because we are diploid, 2 times the number of samples plus 2. And if we do not have a known variance resource, you can set the allele frequency to the average heterozygosity of 0.001. So this slide was provided by one of our developers, and it's meant for those of you who are really interested in the math. And I'm not going to go through it because I don't claim to understand it. If you want to know even more, we have white papers in our GitHub repo. They're just available to anybody that explain all the math for our methods. And that's an open repo. So I'd like to move on into showcasing how the HaplotypeCaller's assembler can help resolve a very large deletion. And this deletion is 120 bases. In the tumor here at top, we see clear evidence via the soft clips that there is some sort of event, and that it is not present in the matched normal here. A traditional pileup caller, such as mutex1, would not be able to call any events here. Also, we couldn't use a discordant read pair caller by taking any rare insert sizes, because all of the insert sizes here are within the normal range. And the HaplotypeCaller assembler, and notice here that I'm using GATK3 mutex2, but it's the same assembler for GATK4 mutex2, is able to resolve this deletion in green, as well as the wild type normal haplotype in pink, plus an artifact in dark pink with insufficient likelihood. So we've just finished mutex2 calling. We have a VCF in hand. None of the variants or the mutations that are called have been filtered. So we're going into now filtering. 2 parts to filtering that we're going to cover today. One part is optional and depends on sequence context dependent artifacts. And the other is estimated cross-sample contamination plus 13 other filters. And we highly recommend that you do the cross-sample contamination. Just never know who's coughed over your samples. You just don't know. So we do recommend that, and that's why it's in a solid line versus the dotted lines. We give our filtering tools these metrics plus our raw variants to get our filtered variants. We're going to focus on the main filtering done by filter mutex calls now. What it does basically is it uses annotations in the info, and this should say info and sample fields in the VCF. It uses all of these to determine, to hard filter based on the annotations. So I'm not going to go into too many details because this part of the workflow is the most in beta. And I know we've just added some new filters recently, and we have removed some. But if you have questions on these, do post on the forum. So currently, the current release of filter mutex call filters for 14 contexts. The filters that you will see in the filter field are listed on the left, and the description that you can find in the VCF header is listed on the right. And I have to say that even if a header lists the filter, for example here, duplicate evidence, it doesn't mean that that filter was used. Some of these require specific annotations that are not emitted by default, and so you'd have to go and make sure you are getting these annotations out during your mutex2 calling to know that the filter is being used. Otherwise, it will list them all, but it may not use them. So in general, just to get a feeling for what the hard filters look for, variants that only occur near the ends of reads are highly suspect. They usually indicate mapping error. Low MapQ reads that support a variant indicate mapping error. Short tandem repeats that are contracted or expanded, these indicate errors. And these errors could be real because polymerase slippage doesn't just happen in a PCR reaction, it happens in our cells. But they could also be errors, and we have filters to filter on 1 to 2 repeats that are lost. If a variant is detected only on one strand of DNA, this is a laboratory error, and we filter for that. Although we do some upfront filtering with the PON and the normal, you can only call on what are apparent before assembly. And so after assembly, the tool double checks to see if a variant is in the panel of normals or in the normal and filters on those, or gives out annotations that filter mutex called and filters on. And okay, yes, the list listed, just wanted you guys to have a complete list of the status right now, but this list contains one filter that filter mutex calls actually doesn't emit, and that's orientation bias. This is a separate tool, as I mentioned. It's called filter by orientation bias. For example, OXO-G, G to T transversions, which is pretty common in sequencing data. And you'll want to run a Picard tool to get the metrics, examine them, and judge for yourself whether you need this additional filtering. So this is just basically the command. So to create a PON, you run mutex2 in a tumor-only mode, but you're providing it your normal sample. And then to do a matched normal somatic calling, to this command you would add your normal sample. And we now recommend that you also include generating the BAM out reassembled reads, because this actually doesn't add that much to the compute. So if you go back to try to generate it, you will be doubling your compute. So just add it from the get-go. It's now actually in our little script to do this. To specify a germline resource, you use this option. And if you do so, don't forget to set the allele frequency of alleles not in resource. And here I'm showing the example for, I believe, nomad exomes. And finally, to specify a PON, you use this last option. Here are the commands for filter mutex calls and filter by orientation bias. And the contamination table that you use for filter mutex calls comes from a new tool in GATK4 called calculate contamination. And presumably, you could have a similarly formatted file from as calculate contamination to which you add your own number if you want to do some filtering, hard filtering based on allele depth. And the order is important. You would do filter mutex calls before you do any filtering by orientation bias. Again, filtering by orientation bias comes second. And here you have to specify the artifact modes for which you want to filter. It doesn't do all of them. You have to tell it what you want to filter on. And here you give it a pre-adapter detail metrics, which is one of many metrics files produced by Picard Collect Sequencing Artifact Metrics. So we've just finished going over short variant discovery. Now we can move on to doing going over the copy number alteration workflow. So these are some resources for you, and we'll just switch to the next talk because there's a different slide deck.